Okay, so I invite you into today's lecture where we'll be looking at a technique which we refer to as recombinant DNA technology. So what this entails is you take two sets of DNA that you obtain from different organisms. So let's say you obtain DNA from a plant and you get some other DNA from a bacteria or even an animal. Then now you combine these two different types of DNA, different DNA from different organisms. So whatever you've gotten from a plant, you combine it with the DNA that you've gotten, for example, from an animal. So what you are creating is a new genetic combination which ordinarily will not occur in nature. So such a process in which you combine DNA from different organisms is what we call recombinant DNA technology. The DNA you create is new, is a combination that is new, is a combination that does not ordinarily occur on its own in nature. So it's DNA that has been recombined, getting DNA from one organism and adding it to another organism. So you are creating DNA that is combined. So to do that, DNA is cut, because remember you are getting it from different organisms. So you have to cut, then you join, and you insert into a host organism. The host organism is the organism that you want to modify the traits. So this technique that we are calling recombinant DNA technology is what is also referred to as genetic engineering. To help us understand this technology better, we'll just quickly look at some terms and how we define them. One of those terms is DNA. DNA is in full is deoxyribonucleic acid. DNA in itself is just a molecule that carries the genetic information in living things. We know, for example, that RNA also carries genetic information, but now the difference between DNA and RNA is on the combination, what DNA is constituted by. So DNA is made up of 4 bases, that is adenine, thymine, cytosine, and guanine. We know for RNA we will not have thymine, instead we'll be having uracil. Another term that is important in understanding recombinant DNA technology is this term that we always call a gene. So what is a gene? A gene is a segment of DNA that codes for a specific protein. So it is a coding region of a DNA sequence, okay, or a functional unit of inheritance. The other term in this case is the term vector. So this is what you use like a vehicle to carry the foreign DNA into the host cell. Okay. So what you use to transfer your DNA of interest from the donor cell into the host cell, that is the vector. Common examples of vectors that we have include plasmids. As microbiologists, you should be knowing that plasmids are small circular DNA molecules that are found in bacteria. So they are there independent of the genetic material of that microorganism. We have also bacteriophages, and these are just viruses that infect bacteria. Then you have the cosmids and the artificial chromosomes. So cosmids and artificial chromosomes are larger vectors, and because they are larger, it means they can now carry bigger DNA fragments. The other term is the word host cell. So when you are combining different sets of DNA, you want that DNA to end up being received by a particular cell. Once it gets into that cell, it should replicate so that it can express the trait. That cell that receives the recombined DNA, that cell is what we call the host cell. Most of the time is an organism, okay? Because you are putting it in a cell of an organism. So the host cell is what receives the recombinant DNA. Then upon receiving, that recombinant DNA will now replicate inside the host cell. The other important concept is the restriction enzymes. So this, to restrict, is to cut. Okay, so restriction enzymes are those ones which are responsible in cutting DNA at specific sequences. They recognize the specific sequences that they cut DNA at those particular sites. So the place where the restriction enzyme cuts is called a restriction site. So restriction enzymes are used for cutting. Then the DNA ligase is an enzyme that helps to join the DNA fragments. So whatever has been cut will be joined together by the DNA ligase. Then the process now of introducing the foreign DNA, the recombinant DNA, into the host cell, that process is what we call transformation. So transformation is the process through which foreign DNA, the DNA that has been created, is introduced into a host cell. So the host cell will be transformed when the foreign DNA has eventually entered it. Another important terminology is cloning or gene cloning. To clone is to make an identical copy. Okay, so this process of gene cloning just means making multiple identical copies of a DNA fragment using a host organism. So you are making many identical copies because a particular segment of DNA will be replicated to create many other copies that are identical to that particular segment that is being replicated. So mostly this creation of multiple copies will be taking place inside the host organism. So it's the machinery of the host organism that is now helping to do this gene cloning. The steps now of recombinant DNA technology. We said that recombinant DNA technology involves picking a certain DNA and adding it or joining it up with a different DNA from a different organism. So it means the first step is to isolate the DNA. You have to isolate, you have to extract the DNA from the organism of interest. If, for example, you want to get a gene from a human being and put it in a dog, then you have to isolate the DNA from the human organism first. Okay, so you have to extract that DNA. The steps of extraction include cell lysis, then there is the removal of proteins and lipids, then you now have to purify the DNA. Then you are not putting the entire DNA into joining in the entire DNA with the DNA of the second organism. You are just getting a segment of the DNA from the donor. So you need, therefore, to cut that segment that you want to introduce. Okay, so the process of cutting, we use the restriction enzymes. So the second step after you've isolated DNA is now to cut that DNA with restriction enzymes. So the restriction enzymes recognize specific sequences. So it means specific restriction enzymes will cut at a specific site. Sasa inamaanisha ukitaka kupata a particular gene, you might realize that depending on where that gene begins and ends, it's possible that you will use a different restriction enzyme than when you are cutting another gene. Okay, so when you use these restriction enzymes, which have the ability to recognize specific sites, they will cut the DNA and either generate what we call sticky ends, these are overhanging ends, or they can cut with resulting into blunt ends. The importance of this second step, where you are cutting DNA with the restriction enzyme, is that it allows you as a scientist to obtain a specific gene from the genome. The DNA that you will have extracted is possibly of the entire genome, but you don't want to insert the entire genome into the host cell. You only want a specific gene. So that's why you are trying to get restriction enzymes that will only cut that specific gene from that particular genomic DNA. Once you've cut and obtained your gene of interest, then the other thing that you want is now to put this gene into a mode of transport. Because you cannot insert the gene into the host by yourself. You need something to help you to insert that gene. So it's at this point that you now find what we call a vector to be very useful. Okay? So the DNA that you've cut, you now combine it with a vector which has been cut using the same restriction enzyme. The restriction enzyme that you use to cut your DNA of interest from the isolated DNA is the same restriction enzyme that you will use to cut the vector with. After you've done that now, you introduce a ligase to help join. Umekata your gene of interest, umekata the vector. So unahitaji kitu ambacho kitakusaidia kuunganisha your gene of interest and your vector. Kitu ambacho kitakusaidia hapo is a ligase. Ligase inasaidia kufunika the nicks. Nicks are those cuts. So once you have a ligase added to the DNA, the gene of interest and the vector, then so long as the gene of interest and the vector were cut by the same restriction enzyme, then DNA ligase will be able to join them together, and at the end of this stage, we'll now be having our recombinant DNA. It is recombinant because it has DNA from a source or a donor organism. That is the organism from which you obtained your gene of interest. You obtain that by using a restriction enzyme. Then, once you've obtained your gene of interest, you need that gene to be put into a mode of transport. That mode is the vector. For the gene to fit into the vector also needs to be cut by the same restriction enzyme. Once you cut the vector and the gene of interest with the same restriction enzyme, you now put them together and you add the DNA ligase, which is sort of the molecular biology glue. Okay, hiyo ndio inaunganisha sasa. Ukimaliza hapo, uko na vector ambayo iko na a gene from a different organism. The vector is possibly a bacteriophage or a plasmid. So at that particular point, you now have recombinant DNA because you've combined DNA from different organisms. The next thing now, we need to use the vector as a mode of transport. Itusaidie sasa kuingiza our gene of interest into the host cell. So that is what we call transformation. The common methods through which the vector together with the gene of interest can be introduced into the host cell include using heat shock. So you take the host cell, ile yenye unataka recombinant DNA iingie ndani yake, then you subject it to some level of heat. You expose it to some level of heat briefly. Okay? That heat is supposed to open the cell membrane. Then hopefully, once the cell membrane has been opened by the heat shock, okay, shock, you take your host cell together with the vector which now has your gene of interest, then you subject the host cell to some brief exposure of heat. The thing is, hiyo heat ambayo inakuwa exposed to the host cell should help ifungue or create a hole on the cell membrane. Then hopefully now the vector can enter through that hole. Another method is electroporation, where you use electric pulses to create pores on the cell membrane of the host cell. Another one is micro injection, where you use tiny needles to directly inject DNA into the host cell. Okay. Once you've done that, you need now to check uone, because possibly not all of the cells will be transformed. Some host cells zitakuwa zimechukua vector imeingia ndani na zingine zitakuwa hazijachukua vector. So mostly there are markers zenye mtu unaweza tumia kujua kwamba hii imechukua the recombinant DNA ama hii ingine haijachukua recombinant DNA. Some of these markers are, for example, the antibiotic resistance genes. If the vector which was used to carry the gene of interest has a gene that makes it resistant against a particular antibiotic, then you can now take all the cells after the process of transformation, then you grow them, you culture them in the presence of that antibiotic. The cell that has taken up the vector will also have taken up the gene that makes it to be resistant against the antibiotic. So then that cell will be able to grow in the presence of the antibiotic. Okay. So once you've selected and identified the cells that have been transformed. Being transformed means they've taken up the recombinant DNA. How you choose these cells, you use selectable markers. For example, antibiotic resistance. So once you've known them, then now you go to the process of gene cloning. So you increase the copies. Okay? You allow now the host cell to replicate so that it can produce many copies of that recombinant DNA. So the product of the gene of interest, if indeed the host cell was transformed, inamaanisha kwamba the host cell will be having that gene of interest. So as cloning happens, that gene will also be expressed. In the process of expressing that gene, then the protein which is encoded by that inserted gene will now be produced. Once that protein is produced, then the function that comes with that gene will now be realized. Okay? Okay, let's have a look at a diagrammatic representation. This is on page 4, okay, where you now have donor DNA. This is DNA that you want to get a gene from, a specific gene from. To get that gene, you digest this DNA with restriction enzymes. The same restriction enzyme you use to digest the donor DNA with is that one that you will also use in cutting the vector, because you want the gene that you've gotten to be put into a vector. Then the vector can help you carry that gene, okay, into the host cell. So you identify the DNA from which the gene that you want is. That is your donor DNA. It is a donor DNA because it is the DNA that is giving the gene of interest. Once you get that gene, huwezi ibeba na mkono, utaiweka kwa vector. It's a vector that will help in carrying that gene of interest into the host cell. That process is what we call transformation. You cut your donor DNA with the restriction enzyme, you cut the vector with the same restriction enzyme. You mix the product of the donor DNA after being cut with the restriction enzyme and the vector after being cut with restriction enzymes. You mix them together, then add a DNA ligase, which is an enzyme that helps to seal, to kufunika, to join the vector together with the gene of interest that you've cut from the donor DNA. Then now, what you've obtained after addition of DNA ligase, the DNA ligase now creates a recombinant DNA molecule. Recombinant because it is now made up of DNA from two different organisms. That recombinant DNA now should be introduced into a host cell. In this case, the host cell is the bacteria cells. So that introduction of DNA into the bacterial cell, hiyo ndio tunaita transformation. Okay? So once the vector together with the gene of interest, what we are calling now the recombinant DNA, once it is introduced into the host cell, then that host cell is now transformed. After that introduction, then now we do cloning. We allow that particular bacterial cell that has now been transformed to create many copies of itself, to create clones. A clone is a copy, okay? An identical copy. That's why now you end up with many copies of the same recombinant DNA because the transformed host cell has been allowed to undergo replication and expression. Okay, so at that particular point, then the entire process of recombinant DNA technology is complete. Okay. So you might be wondering, why are we dealing with recombinant DNA yet you are microbiologists in the making? The answer lies in the importance or the critical roles that microorganisms play in this recombinant DNA technology. So bacteria and yeast are very essential. They play critical roles in recombinant DNA technology, mainly because they grow very fast. So it means you can have so many clones within a very short time. 20 minutes, for example, for E. coli, it has divided. So if you leave it overnight, you'll find tomorrow you have many clones. Then it's easier, it's cheaper to maintain microorganisms. Okay, the cost, for example, you need media, you need an incubator, and that's it. Okay. Then their DNA, their genes, their chromosomes, and their genomes are smaller, so it's easier to manipulate them. Okay. We talked of plasmids being important vectors in this process. So because in some bacteria you naturally find plasmids, then it means such bacteria are now offering a natural source of vectors. There are also ways in which microorganisms can take up foreign DNA readily. Okay? So when a cell takes up foreign DNA, then we say that cell is competent. Okay? So microorganisms can readily take up foreign DNA. Okay? Then because of the easy and shorter time span, then these microorganisms can produce large amounts of proteins. So for example, if you want to produce insulin, it's easier to produce insulin using a microorganism, using a bacteria. They do it in E. coli. You can have vaccines also produced by microorganisms. It is easier than if you want to put it in a plant or an animal. Okay. So we've talked about competent cells. When a cell has the ability to take up foreign DNA from its surrounding, then that cell is said to be competent. That ability for the cell to take up DNA from the foreign DNA from the surrounding can be attained either naturally or after that cell has been treated. So competence is now the ability of a cell to take up foreign DNA. So this competence is needed because if cells cannot allow DNA to enter, then we cannot have recombinant DNA being formed. Okay? You need the recombinant DNA, but it has to be put into a host cell. It has to transform a host cell. That process needs the host cell to be competent. Okay? So the competence is important because it is through a cell being competent that that cell can be transformed. Then after the cell has been transformed, then cloning and expression is possible. Okay. So cells can be competent either naturally. Okay. So it means nothing is done to them, but naturally such cells can just take up foreign DNA. Examples of bacteria which are naturally competent are Bacillus subtilis, Streptococcus pneumoniae, and Neisseria gonorrhoeae. Okay, so such bacteria, because naturally they are capable of taking up foreign DNA, it's been realized that they have special membrane proteins that allow them to take DNA naturally. Okay. Other cells that are not competent by nature then should be made competent artificially. Okay. Artificial competence in cells can be attained through two main methods. One is a chemical method. In this chemical method, where cells are made competent chemically, the cells are treated with calcium chloride. So the calcium chloride will weaken the cell membrane. Okay? You take up a host cell, put it in the presence of calcium chloride. The thinking is the calcium chloride needs to tamper with the cell membrane so that the cell membrane, being the first stop, the first place that helps the cell not to take anything in unless that selective permeability. So for you to interfere with that selective permeability, then you bring in calcium chloride so that you weaken it. You make what is otherwise a strong protective organ, part of the cell, not to be such strong. You weaken it. Okay? So with that, then you end up now creating pores. And it is through these pores that DNA can now be sucked into the cell. How were pores created? The pores were created because there was a chemical. This chemical is calcium chloride. Calcium chloride weakened the cell membrane. Weakening the cell membrane created mashimo, or the pores. Then through those pores, now DNA can enter the cells. Another way in which cells can be made competent artificially is electroporation. Okay, electroporation. Electroporation means you subject these cells to a strong but quick electric pulse. You subject them to some sort of electric shock. So the thinking is that shock that you are subjecting the cells to, hiyo ndio itatengeneza mashimo on the cell membrane. Then kupitia kwa hayo mashimo, DNA can now enter. Once DNA enters, then that cell is said to be competent. But it was not competent naturally. So it had to be made competent using an artificial means. Okay. Okay. So in which fields then can recombinant DNA find application? The answer is in several, but we'll just list a few. One, in medicine. We have, for example, the hormone insulin being produced using recombinant DNA. We have the production of the human growth hormone. You have the production of the blood clotting factors. There are several vaccines, okay? And we also have gene therapy. So recombinant DNA can be applied in medicine to produce various hormones like insulin, human growth hormone, blood clotting factors. It can also produce vaccines, among other things. The second area is in agriculture, where we have now the creation of genetically modified organisms, in this case, crops. Such genetically modified crops will have some improved traits. So you have things like pest resistance, herbicide tolerance, drought and disease resistance. Then you can also enhance certain vitamins in some of these crops. Okay, so you can enhance the nutritional value of some of these crops. Another area is you can apply recombinant DNA in industry, where you can use it to produce detergents, you can use it in the production of biofuels, you can use it in the food processing. In the other area is in research, where you want to study, for example, the gene function. So you create a mutation, you stop the expression of a particular gene, or you lead to the expression, then you see how that gene functions. Okay. Does recombinant DNA have any advantage? The answer is yes. Okay. So one is the high precision, which means genes can be manipulated accurately because you target a particular gene, you cut that gene. So it's not guesswork. Lazima ujue which gene you want. So that is the precision. Then because you are putting the recombinant DNA in a bacteria, for example, which reproduces so fast, the second advantage then is the rapid production of biological products. Within a very short time, you end up having any biological product that you wanted being produced rapidly. Okay. When you've genetically modified crops, you've modified a particular crop possibly so that it can withstand conditions or pests, then you end up improving the yield in agriculture. So that is an advantage. It can also be used in the treatment of genetic diseases. It can also enhance food quality. On the other side, there are also disadvantages that come with recombinant DNA. One is the ethical concerns. When you are genetically modifying an organism, there are people who think that there could be issues about safety. You are creating something that might not be safe. Okay? Others also think that it is only God who can create genomes. So if you are tampering, if you are creating a new combination, then you are playing, you are acting like God. Okay? There are other people who think that through this process of recombinant DNA technology or genetic engineering, it could lead to the genetic modification of human beings. Okay? But of course, it's not permitted to genetically modify a human being. Another disadvantage is environmental risk. You have a genetically modified crop. What if it crossbreeds with a species that was not genetically modified? So then what are you creating? Okay. So there is that risk that in the process of this, because you remember the selectable markers was the antibiotic resistance, what if through the recombinant DNA technology we end up spreading markers that are associated with antibiotic resistance? Another thing is the high cost and technical expertise that is required for genetic engineering. You need advanced equipment. You need very high levels of training, and you need strict conditions in the laboratory. So there is a cost component, then there is a technical expertise level that is required. Okay. The other thing is there are the unintended effects. What if the gene behaves in a way that was not predicted? You thought that when you put the gene, it will lead to resistance to that particular pest. But ukiweka hiyo gene, then ifanye tu something that was not intended, ni kitu ambacho hakikuwa kinatarajiwa. Okay? Then the last thing is public resistance. You must have heard many people, churches, talking about GMOs. Okay, so such people remain very skeptical. They don't want to trust GMOs. They don't want to embrace them. Mostly, the skeptical nature of most people about GMOs is largely due to either they are misinformed, their cultural beliefs, or religious beliefs. Okay, you have had some churches come out very strongly warning their congregants not to take up GMO. Okay, so there is an aspect of public resistance, and that is mainly because either people are not properly informed, people have religious beliefs that are against GMOs, or they have cultural beliefs that are also against GMOs. So just to summarize, recombinant DNA is a powerful tool which allows scientists to combine DNA from different sources. Then, by combining DNA from different sources, they end up creating organisms that have new abilities. Okay. Microorganisms, they form the backbone of this technology, this recombinant DNA technology, because one, they grow very fast, they are genetically simple, and they produce proteins efficiently. For recombinant DNA technology to happen, we need cells that are competent. Cells can be competent naturally, or that competence can be artificially induced. For they to be competent, it means they are able to take foreign DNA. Once the cells have taken up foreign DNA, then they end up being genetically transformed. Okay? So despite the many applications associated with recombinant DNA technology, there still are various aspects that are raised, mainly dealing with ethical, ecological, and safety concerns. So because of that, then, we must carefully manage recombinant DNA technology. Okay? So we've gone through this topic of recombinant DNA technology, which is also called genetic engineering. Have a listening. Should you have a question, reach out. Okay? Otherwise, thank you very much, and I look forward to further engagements. Asante.